Welcome all. Today in this video we are going to discuss the functional histology of the spleen. Myself Dr. Ankit Khandelwal, MBBS MS in Anatomy, and this is my YouTube channel Anatomy Explained. Don't forget to subscribe to the channel. Moving to the topic, that is the functional histology of the spleen. In the previous video we did about the gross anatomy of the spleen. We talked about the location, the relations, the ligaments, the vascular supply. So we know by now that the spleen is supplied by the splenic artery, which is a branch of celiac trunk, and the venous drainage is by the splenic vein, which forms the portal vein behind the neck of pancreas. The link of that video will be there in the description box below. So in this video we are dealing with the functional histology. So for that we need to know the basic functions. So as we know that the spleen is the largest aggregation of lymphoid tissue in the body. So its first major function would be the production of the immunological response to the blood-borne pathogens. So that is one major function of the spleen. And the second major function of the spleen is to remove the particulate matter, or you can say remove the old or denatured blood cells, particularly the RBCs over here. Old or denatured RBCs, blood cells like WBCs or platelets. In the fetus also it has a role. In the fetus it has one of the major role, and that is your blood production, blood cell production, known as hematopoiesis. That function is mainly observed in fetus. In adults also that could be that function could be come again, but only in few of the special diseases. So we will be focusing mainly on these two functions, and let's learn the histology of the spleen in this way. So the first major function was to produce the immunological response for the blood-borne pathogens. Now if we know about the spleen, then the spleen has an outer covering, that is a capsule, and through this capsule there will be few indentations which will be going inside the spleen. Those are known as basically our trabeculae. So these will be our trabeculae. These are just the indentations from the capsule. So these will be our trabeculae. Now these indentations are for what? They are basically meant to go inside the substance of any organ, so that the passage of the vessels, the nerves, the lymphatics would be taken from the outside surface of any organ to the deep inside. So that is the role of trabeculae. So what was the first function? If we remember, the first function was the production of the immune response, to the blood-borne pathogen. So what do we need for that? We need a blood vessels should be going, or the arteries should be going inside the substance, and around those arteries there should be immune cells. So that is the function of here. So through these trabeculae there will be few of the branches of splenic artery which will be enter, mainly the segmental arteries and its branches, and those segmental arteries are basically end arteries. So these will be, so just imagine an artery going inside the substance of the spleen. Now around this artery there will be lot of immune cells which would be surrounding the artery or its branches. So that is actually the inside histology of the spleen. This part of the histology of the spleen is known as the white pulp of the spleen, and that constitutes around 5 to 20% of the whole substance of the spleen. So white pulp is what? White pulp will be having a central artery which is actually located eccentrically. We'll see it in the slide, but major function is that artery will be surrounded by immune cells, both as a T lymphocytes, as a B lymphocytes, as the macrophages, and that area is the white pulp. So if you look at the histological picture, it appears like this. This is what? This is the outside, that is the outside capsule. Okay, and as it is a section, so you can see few of the trabeculae over here. These will be few of the trabeculae. Okay, this area over here, that is showing you one area or one white pulp over here. Okay, one white pulp. Now that is a stain, so the color are but bound to be somewhat different. Okay, but in each of this white pulp, what you will be able to see is that you will having an artery over here, you will having an artery over here. Okay, you will having an artery over here and there. So this will be a branch of that segmental artery which is surrounded by the immune cells, the T lymphocytes and the B lymphocytes and macrophages. That area is known as the white pulp. Now let us see the details of it. This is the white pulp, constituting around 5 to 20% of the histology of the spleen. So one function, and this is the location of it. Now if you look into details of it, this would be a branch of the splenic artery that is entering, and this branch of the splenic artery would be giving off few arteries, few branches which will be like perpendicularly located, or they will come out as a perpendicular branch to this main segmental artery. These are known as penicillar arterioles because they are now smaller in number. These are known as penicillar arterioles. At the tip of the peninsular arterioles, you have because these are going to decrease in size, you have a small bulging which is known as the sheathed capillaries. These are known as sheathed capillaries. So these are known as sheathed capillaries. Now the property of sheathed capillaries is that there is no endothelium present in the sheathed capillaries, and these are surrounded by macrophages. So here if we try to make that, around this you will have a lot of macrophages surrounding this part. We can make it like this also. There will be a lot of macrophages surrounding these sheathed capillaries. Point number 1. Okay, so you have the branch artery going inside, and that will give one of the branches known as peninsular arterioles. At the tip of that you will have the sheathed capillaries, which are not having any endothelium, but yes, they will be surrounded by macrophages because the blood cells have to come out of this. So as the main artery was going inside, it is also surrounded by a periarterial lymphatic sheath. The main artery that is going inside from the trabecule is surrounded by, very important, you have to keep that in mind, very importantly, this is known as your basically made up of T cells. In T cells, they are basically the helper T cells. They are known as periarteriolar because they are around the arteriole. Periarteriolar lymphatic sheath, also known as PALS. So you can see the first function was production of immune response to the blood-borne pathogen, and as the blood vessel is entering inside, it is surrounded by periarterial lymphatic sheath, mainly made up of T helper cells, and around the branches of it, and then from the branches at the ends of it, you have the sheathed capillaries covered by the macrophages. Not just that, even over here you will have a group of B cells also over here. They will be forming aggregations around the terminal ends of these branches. There will be aggregations of lymph tissues, or we can say lymphoid tissues, which are mainly formed by the B cells. So these are also surrounded over here. These are also surrounding this whole area. That is your whole inside histology of the white pulp. So that is serving this function, production of immune response. So if there is any pathogen inside this artery, let us try to make over here artery little bit thicker. If there is any pathogen inside this artery or arteriole, that will be taken care by this T cells, the B cells, and the macrophages. Okay, so that is your white pulp, which constitutes around 5 to 20% of the whole histology of the spleen. Okay, now from here what happens, from the trabecule you will also have few veins, because the vessels they pass through trabecule. So yes, from here there will be few veins over here, we can call them as sinusoids, and they will be combining over here, and they will be again draining back where, through this trabecule as the branches of the splenic vein. So in this, the direction of blood flow will be like this, and the arteries, the direction of flow will be like this. So but first of all, the function, immune response, white pulp, 5 to 20%, T cells, B cells, macrophages. Let us move to the next function. The next function of the spleen was to remove the particulate matter, was the removal of all the particulate matter, old denatured RBCs. So we write the particulate matter or RBCs, but not the normal one, but the old or which are deformed or denatured RBCs. Now how is the spleen going to perform this function? Now again, if we try to do the same thing, we try to make a capsule over here, we try to make the trabecules over here. Okay, so now we already made that vessel, that artery part, so we can simply make it like this. This was the artery, and these were all its branches which were coming off. They were actually covered by all the cells of immune response. And from this trabecule part, what we have, from the other part, we have this venous sinusoids. Now venous sinusoids are what? They are actually sort of dilated capillaries. They will be all over here, they will be combining, and they will be forming what? They will be forming, combining, and they will be forming the veins over here, some branches of air over here also. So these are your venous sinusoids. Now look at the function, removal of the particulate matter or the old denatured RBCs. Now what comes from this artery part? Let us take this green color. Now what was coming, we had that sheathed capillaries, and from that the blood cells started to come out. The blood cells will start to come out from here. Okay, they have to travel through this whole substance of the spleen, and then they will try to enter into this venous sinusoids, because that is a property. artery brings the blood and veins take the blood. These venous sinusoids, what are they having? They are having few of the endothelial cells obviously as their lining. But the special thing over here, special property, is that first of all these endothelial cells, they look like barrels of wooden barrels, so they are called as stave cells. That you have to remember, that could be one question, stave cells. Plus in between the endothelial cells you have a few small slits present, so there are few small slits which are present between the endothelial cells. So when this any particulate matter or any RBC has to pass from the artery to the venous sinusoid, it has to actually fold somewhat and it has to squeeze its way through the venous sinusoid. So for any cell to pass through this between the endothelial cells, that cell need to be fresh and active, and normal cell only that can pass through it. So whenever the cells which are old or denatured or which are not flexible, they want to be able to pass between the slits, and therefore they are trapped over here, because all that space is also surrounded by macrophages. Now what is that space? Now this whole thing which is occurring as a removal of the particulate matter, this was our, if we recall, the white pulp, which formed the 5 to 20%, and whatever is left over here, whatever is this area, that is basically made up of our red pulp. So red pulp forms up to 90% of the total this histology of the spleen. Now from the artery end, where the cells were coming out, between that arterial end and this endothelial lining of the venous sinusoid, that is your stave cells, you have this layer which is actually formed by our what we can say as the type 3 collagen or the reticular fibers. So over here we have a lot of reticular fibers, and not just reticular fibers, we will have fibroblasts, we will have macrophages, few of the immune cells which are present over here. So this actually is forming what? This actually is forming the splenic parenchyma over here. So when the blood has to pass from artery to the venous sinusoid, it has to pass through this splenic parenchyma, which in 2D image looks like cords. So they are known as splenic cords. Or also known as cords of Billroth. They are also famously known as cords of Billroth. Okay. So when a cell has to pass, the blood cell has to pass, or any particular matter has to pass from artery to the vein, it has to pass through this parenchyma or these splenic cords or cords of Billroth. So these splenic cords and this venous sinusoid, they combine, they form the red pulp. So what is this red pulp over here? Try to understand, because we are doing the functional histology. It is basically made up of that splenic cords, or known as cords of Billroth, and your venous sinusoids. Now particularly for interest over here, the splenic cords, as we said, cords of Billroth, they occupy 70% of the red pulp by volume, and venous sinusoids, they only form 30% of the red pulp by volume. Point number one. Second thing is the splenic cords are not just that reticular or type 3 collagen, they are also fibroblasts, they have macrophages and all. Second thing, if you remember, these venous sinusoids lined by endothelial cells, they were known as stave cells. Third thing is, the appearance of the red pulp is the appearance of like a Swiss cheese pattern, also known as Swiss cheese pattern. Now what does that mean? We know that when the cheese is formed, it's formed by this fermentation. So let us see that part. First of all, let us see this over here. This was our white pulp which we saw. So this was one white pulp, this was one white pulp. I'm writing WP. Whatever is between them, this whole structures over here, they are our red pulp. You can see over here a lot of staining of the venous sinusoids. So whatever is not white pulp is your red pulp. Therefore red pulp is up to 90% maximum, and this white pulp forms 5 to 20%. Now as we know that red pulp was formed by what? Splenic cords or venous sinusoids. So in a 2D image they look like cords. The parenchyma, it looked like cord, but if we look at a 3D image, they look like a Swiss cheese. Now what does that Swiss cheese means? You look at a figure over here, just a cheese over here, and of cheese is formed by fermentation. So these small holes, they are actually representing the venous sinusoids. They actually represent venous sinusoids. All that cheese material is actually your the splenic parenchyma, not the renal, but the splenic parenchyma, or we can say the splenic cords. So this cheese, or you can understand, majority of, therefore it constituted 70% of the red pulp, and venous sinusoid constituted 30% of the red pulp. So that is how the red bulb is arranged, and this is how the second part of function is done, removal of the particulate matter. Because if suppose this green cell, if I try to make it again, it is coming out from this RT line after all that immunological response. It has to make its way throughout the splenic cord, then it has to reach over the ends of the venous sinusoids, it has to fold on itself, then only it will be gain entry inside the venous channel, and then from here the blood direction through the trabeculae outside, they all combine in the form of splenic vein. So these are the two main functions which are subserved by this histology of the spleen, and therefore what we saw over here as the histology of the spleen. Now the third thing which is asked in the spleen and which we have to understand is about the circulation of the spleen. Not the gross circulation, but over here the micro circulation. Histology is micro and not micro circulation. There are two types of micro circulation which are called in spleen. They are known as open circulation. They are known as open circulation, and the other one is known as closed circulation. Other one is known as closed circulation. Let us try to understand the differences between both of these circulations. So let us try to again make up this capsule over here, and from the capsule let us make one big trabecule. From the trabeculae we can start to making, okay, this is the artery and its branches which are coming up, okay, and these are, say, it's a few of its branches which are going up. Clear? And from the other side we know that, okay, this would be the venous sinusoids. They will be actually over here, and they will be combining, and they will be forming this branches of splenic vein, and they will go in this direction, and the artery was coming in this direction. Okay, that is what we know. This area around the main artery, that was our white pulp, and this whole area was our red pulp that we already saw. Now what are these two types of circulation? These two, now we can make a little bit of, say, this splenic cords also. So if we try to make the splenic cords over here, that is the splenic parenchyma, this would constitute some part, we can say, of the splenic parenchyma, constituting all of the reticular fibers, fibroblasts and all that. Now coming back to our blood cells, now if we make the blood cells like this, now one type of circulation is when the blood cells are coming out of these branches or sheathed capillary or peninsular arterioles, they will traverse through this whole splenic cord normally, and then they, if they are normal enough, they will enter into this splenic vein and they will go in this direction. This type of normal circulation is known as our open circulation. Okay. Now second type of circulation is when the blood cells, they come out from this capillary part, sheath capillary, and they directly enter into the venous channel without the intervening splenic cord. So that type of circulation is known as closed circulation. That type of circulation is known as closed circulation. Open circulation, you have to pass through splenic cord, just remember that. Closed circulation, there is no intervening splenic cord, so it directly passes. Now there is not much of difference over here, because if they are also in a closed circulation, even then they have to pass through few cells, because remember the capillaries were surrounded by macrophages, and inside that little part of distance which they have to cover through the venous sinusoids, again there will be few immune cells over there. But still, this is a type of, theoretically there are two types of circulation, an open type of circulation and a closed type of circulation. So this was our functional histology. I hope you liked it. So functional histology is the best way to understand histology, and if you liked it, then don't forget to subscribe to the channel Anatomy Explained. That's all. Dr. Ankit Khandelwal, Anatomy Explained. Thank you.